The William Guy Forbeck Research Foundation is proud to present Understanding Cancer Cell Plasticity.
For decades, cancer research has been driven by a singular mission: mapping the human genome to hunt down specific cancer-causing mutations, like KRAS and P53. We developed precise targeted therapies designed to hunt and destroy cells harboring those specific genetic errors. Yet, time and time again, tumors find a way to survive the chemical assault and return. When researchers looked closer at how these resilient tumors were evading treatment and spreading, they found something unexpected. The cancer cells weren't altering their underlying DNA sequence to survive. The genetic code was exactly the same. If the DNA sequence itself remains unchanged while the cancer adapts, our decades-long fixation on genetics has left us with a critical blind spot. The tumors are using an entirely different survival mechanism to beat our best drugs. To see how they pull this off, we have to look at how a healthy body builds itself. Through a process called differentiation, generic cells lock into specialized roles. They become the tissue that makes up our eyes, our hearts, or our lungs. Once a cell is differentiated, it adopts a highly specific physical structure to do its job. It essentially locks into a permanent, lifelong identity. But cancer has a way to hit the undo button. Its ultimate survival tactic is reversing that specialization. It discards its physical structure and abandons its fixed identity entirely. This means if we want to understand how a tumor outlives our therapies, we have to stop looking exclusively at its genetic code and start looking at how the physical state of the cell is actively shifting. Scientists call this ability to change state cellular plasticity. And while it makes cancer incredibly deadly, the mechanism itself is actually a normal, healthy part of daily organ function. Take a condition called Barrett's metaplasia. Every day, the cells lining the human esophagus might face constant minor injuries from acid reflux. To survive the acidic environment, esophageal cells protect themselves by changing their makeup to act like intestinal cells. Crucially, they achieve this shapeshift without touching the core gene sequence. Instead, they alter gene expression, turning the volume up and down on specific genes. Plasticity is a survival trait hardwired into our developmental code to help normal tissues adapt and survive physical trauma. Cancer cells hijack this developmental flexibility. When threatened by inflammation, a new internal mutation, or a targeted medical therapy, it reawakens the plasticity program to escape the threat. This is especially dangerous in carcinomas. These arise in epithelial tissues where cells are tightly interlocked to form rigid borders. To metastasize and spread, a cancer cell uses plasticity to change its physical makeup, dissolving those rigid borders to break free from the wall. Once detached from the epithelial grid, these shape-shifted cells can migrate through the body, arriving at distant organs to seed entirely new tumors. Corrupted cellular plasticity is the mechanical engine driving the disease. It allows the tumor to slip past our strongest drugs and provides the exact mechanism required to spread throughout the body. Defeating a threat this dynamic requires an entirely new approach to how we organize medical research. Because plasticity is a shared biological trait, it spans multiple tumor types, from prostate to lung to pancreatic cancer. Historically, specialists have studied these organs in isolation, rarely comparing notes. That structure is finally starting to crack. Massive initiatives like the Cancer Grand Challenges and the Forbeck Forums are bringing developmental biologists, pathologists, and organ-specific researchers together to merge their findings. Effectively targeting drug resistance requires dismantling decades of academic silos to identify the overlapping biological themes shared by different cancers. Right now, scientists can observe these shifts. They can define the specific molecular changes happening inside a plastic cell, but they don't yet have the pharmaceutical tools to intervene. The ultimate medical objective is to develop a new class of drugs designed specifically to block a cell's ability to change its physical makeup. The goal is to trap the cancer in its current state. If cross-disciplinary science can figure out how to freeze these cells in place and strip away their developmental disguise, our existing arsenal of targeted therapies can finally finish the job.